Constituent factors observed in messenger RNA vaccination. Terms, principles, and mechanisms of action. Pause text and illustrations for detailed study. Refer to the previously posted constituent factors observed in viral diseases, parts 1 through 5. Virus spike protein structure and life cycle illustrations, and characteristics of novel genetic variants possessing viral envelope spike proteins for data relevant to this presentation. The conventional definition of vaccine describes a suspension of attenuated or killed bacteria or viruses, including their antigenic protein or peptide derivatives, administered for the amelioration, prevention, or treatment of infectious diseases. The goal is to induce immunity. Vaccination consists of an immunizing procedure in which a vaccine is administered. Novel messenger RNA vaccines transfer synthetically produced RNA into human cells. This mRNA protein molecule functions within cells, genetically programming them to produce the foreign antigen produced by the pathogen or cancer cell. Cellular or humoral adaptive immune responses can be subsequently stimulated, as a result, rendering the body capable of specifically destroying the pathogen or cancer cell. Protein-based vaccines deliver immune system stimulating antigens in forms such as attenuated or killed inoculum, subunit vaccines, as well as virus-like particles. Immunogenic gene-based vaccines bear the genetic instructions for host cell antigen production, closely mimicking specific natural infection. The mRNA vaccine specifically encodes the SARS-CoV-2 spike protein the virus utilizes to bind and fuse with human host cells. This genetic material instructs the cells to produce the S protein, to which they produce a protective antibody response. RNA transcription illustration. Messenger RNA interaction illustration. mRNA never enters the cell's nucleus. Peptide synthesis illustration. Ribosomes translating messenger RNA. Ribosome translation illustration. A ribosome is a minute entity composed of RNA and associated proteins found in the living cell cytoplasm. Ribosomes bind messenger RNA and transfer RNA for polypeptide and protein synthesis. Messenger RNA bears the genetic code utilized by cells to synthesize proteins. Messenger RNA specifically dictates to the cells which proteins they will produce. Two essential processes must occur within cells for a protein to be produced based on the genetic information contained in DNA. This data encoded in DNA is transferred to mRNA during the process of transcription. The mRNA vaccine, encoding the viral spike protein within the lipid nanoparticle, enters the host cell, where the protein is translated in the ribosome, where it is broken up into smaller peptides by the proteasome, or transported via the Golgi apparatus to the cytoplasm, where ribosomes translate the mRNA into protein in this process of translation, whereby functions are elaborated at the cellular and tissue levels. The peptides are then presented in an MHC class I complex on the cell surface. The complex is recognized by CD8 plus T cells, resulting in cell-mediated immunity. Spike proteins outside the cell may be assimilated by various immune cells and be broken up into peptides by the endosome in a complex MHC class II protein for presentation onto the cell surface. Recognition by CD4 plus T cells resulting in B cells occurs, producing antigen-specific antibodies to SARS-CoV-2 in this antibody-mediated process. Memory B cells associated with immunoglobulin G are believed to be induced post-vaccination, functioning in longer-term immunity. MHC tetramer binding to T cell, left side of illustration. MHC molecule on the surface of an antigen-presenting cell binding to T cell receptors, right side of the illustration. An MHC tetramer contains 4 MHC peptide complexes that can bind to receptors on an antigen-specific T cell. A fluorescent molecule is attached to the tetramer for analysis of T cells. MHC molecules are expressed by most cells in the body and present peptides that T cells can recognize and respond to. Peptide binding for class I and class II MHC molecules, showing the binding of peptides between the alpha helix walls upon a beta sheet base. The difference in binding positions is shown. Class I primarily makes contact with backbone residues at the carboxy and amino terminal regions, while Class II primarily makes contacts along the length of the residue backbone. The precise location of binding residues is determined by the MHC allele. The major histocompatibility complex comprises a genetic locus on vertebrate DNA of polymorphic genes coding for proteins essential to the adaptive immune system. The MHC functions in signaling between lymphocytes and antigen-presenting cells, as well as the rejections of incompatible grafts between individuals. Cells interact effectively with other cells sharing an MHC haplotype. This system allows large immune system cell proteins to recognize compatible or foreign proteins. MHC molecules mediate interactions of white blood cells with other white blood cells or body cells. Of the MHC class I, II, and III subgroups, the 2 types of genes coding for MHC class I and II molecules function in antigen presentation. MHC class I encodes peptide binding proteins that select short amino acid sequences for antigen presentation, as well as molecules that support antigen processing. MHC class I expression involves the alpha amino acid chain comprised of CD8 receptor ligands on cytotoxic T cells and inhibitory receptors on natural killer cells. MHC class II encodes peptide binding proteins as well as proteins functioning in antigen loading onto MHC class II peptide binding proteins. MHC class II expression involves alpha and beta amino acid chain ligands comprising the CD4 receptors on helper T cells. The mRNA COVID-19 vaccine involves CD8 plus T cell mediated MHC class I pathway immunity, as well as antibody mediated CD4 plus MHC class I pathway immunity. mRNA vaccines are designed to harmlessly imitate the capability of the coronavirus to trigger the body's immune responses to this infection and give rise to these 2 forms of immunity. RNA vaccine mechanism of action illustration. The genetic sequence of COVID-19 was isolated and posted online by scientists at its Wuhan, China place of origin on January 10, 2020. Gene-based vaccines bear the genetic instructions for host cell antigen production. Such production also occurs in the case of natural infection. The surface spike protein the virus utilizes to bind and fuse with host cells comprises the antigen. In the case of COVID-19 mRNA vaccine, it provides the host cell's genetic material containing the instructions as to how to specifically produce the spike protein to which they will produce a protective antibody response. The vaccine encodes the surface spike protein receptor binding domain. Binding antibody responses to full-length S proteins as well as the receptor binding domain occur. Substantial neutralizing antibody responses also take place. Binding and neutralizing antibody levels are also induced. Rapid seroconversion for binding antibodies occurs. The magnitude of immune responses are vaccine dose dependent. Resulting CD4 plus and CD8 plus T cell, as well as binding immunoglobulin G antibody immune responses become measurable. IgG production is mediated by B cells. These responses are protective against viral replication. This immunogenic vaccine produces viral binding and neutralizing antibody levels similar to same observed in the serum specimens of convalescent COVID-19 patients whose bodies exhibit an immune response to the virus. The two-dose mRNA COVID-19 vaccination schedule produces immune responses also influenced by factors such as disease severity, patient age, and time since disease onset. Cell-mediated and humoral immune responses have been associated with vaccine-induced protection against antigenic challenge or rechallenge after SARS-CoV-2 infection. An individual strand of RNA is composed of 4 nucleosides. A hybrid form of mRNA for vaccination was formulated to mimic the mRNA naturally produced by human host cells that does not signal for an adverse immune response, as may occur in the presence of a synthetic foreign form of mRNA. This specific genetically modified and purified non-toxic vaccine-grade hybrid mRNA is capable of incorporating itself into host cells for the production of spike proteins without adversely alerting the body's natural defenses and triggering dangerous immune reactions, providing for a protective coronavirus immune response. This is necessitated by the fact that free mRNA rapidly breaks down in the body, resulting in a time constraint that may attenuate the possibility of a desired immune system effect. This complication can be circumvented incorporating the specific hybrid mRNA into a stable fatty capsular lipid nanoparticle that facilitates its immunogenic delivery into the host cell. The presence of a naturally folded perfusion conformation of the spike glycoprotein to the immune system from a specific mRNA template allows for host cell antigen production as well as immunogenically sufficient antibody responses to SARS-CoV-2. mRNA is broken down and eliminated by cells once its genetic instructions have been elaborated. mRNA vaccines are not capable of producing infection since they are not produced using live COVID-19 virus. An appropriate cold chain temperature shipping, delivery, and storage capability from the vaccine point of manufacture to the clinical site and time of vaccine administration must be maintained to preserve the efficacy of the inoculum to the patient. It has been postulated that COVID-19 vaccines may confer neutralizing antibody immunity subsequent to 2-dose vaccination for about 3 months or about 8 months for 1-dose vaccination. As of the completion of this presentation, COVID-19 mRNA vaccination has recently begun using vaccines manufactured by various pharmaceutical companies. Measurable levels of vaccine-induced COVID-19 neutralizing activity were observed. The degree of acquired long-term immunity conferred has yet to be ascertained. New presentations coming soon.